In this video, we're going to discuss experiment 8, which is our synthesis of nylon 6,6. So nylon 6,6 is a polymer, and it is synthesized through what we call an interfacial polymerization. And so interfacial polymerization is a type of step growth polymerization reaction. And so I've got a nice depiction up here of what a step growth polymerization will look like. And so when we're talking about polymerization reactions, we're taking smaller units, which are called monomers. And so those are represented by these circles over here that are not filled in. So we've got our small monomers, and we begin to attach monomers to one another to make our polymer. And so we can see parts of our polymer which are represented by the black filled in circles here. And so in a step growth polymerization reaction, we have a mechanism where we are first forming these sort of bi or multifunctional monomers. And so those are forming our dimers. So we can see two monomers combined together to form dimers. We've got a few dimers represented over here. And then we can see those dimers, some of them grow into trimers. Sometimes we end up getting some oligomers here. And then over time, these all begin to kind of polymerize with one another to form our longer polymer chain, which we can kind of see here. We've got some long polymers. And so the interfacial polymerization reaction that's taking place in our nylon synthesis is a type of step growth polymerization when the polymerization is happening at the interface of two immiscible phases. And usually these are liquids. So what we are going to have is we are going to have adipoyl chloride and dichloromethane, and that is going to be reacting with hexamethylene diamine. And the polymerization reaction happens at the interface of those two immiscible solutions. And so a few examples of materials that can be formed by interfacial polymerization are ultra-thin films, along with nanocapsules and nanofibers. These are important materials that have become very important in the chemical community as of late. So let's take a look at our nylon 6,6 reaction scheme. And so we are going to start this with adipic acid. And so adipic acid is a dicarboxylic acid. Gotta fill all those lone pairs in so we can keep track of our electrons. So we've got our adipic acid, and we're going to react this with thionyl chloride, SOCl2, dimethylformamide, DMF, and we're going to use a catalytic amount of DMF. We're going to do this in dichloromethane, 50 degrees Celsius for approximately 90 minutes. And this first reaction is essentially going to convert our carboxylic acids into our acyl chlorides, or acid chlorides. And so if we don't have any sort of molar quantities, we can assume that we have an excess of each of these reagents. And so at this point we can take our what is now adipoyl chloride, and we can react this with our next reagent. And that's going to be our hexamethylene diamine. We're going to do this in the presence of sodium hydroxide, which is aqueous. Because that's aqueous, we can always assume that we have water around as well. So when we take a look at the mechanism, we're going to have sodium hydroxide and water both present. We'll go ahead and add our lone pairs up here just to keep things consistent. And so ultimately what we've got here is we've formed an amide bond between what was our acid chloride here and our amine here. So now we've formed an amide. So we've lost essentially HCl, so we can put that here if we want. I'm going to put that in quotes because it's not going to be seen as HCl exactly, but essentially we've also produced HCl in this reaction as well. There are a couple of things here that are important for us to note. So one thing that we might notice, we have one equivalent of each of these, so let's make sure we put that here. So if we're showing just one step of this polymerization reaction, we'll assume just one equivalent of each of these, and that's going to give us our one amide bond. Now we've shown here that we've got a carboxylic acid on the left-hand side of our molecule. Now why is that? Because in this step we take our acid chloride, which is on the left-hand side of the molecule. If we're only reacting with one equivalent of our hexamethylene diamine, then we can only form an amide bond essentially with one of our acid chlorides. And so when we've got our sodium hydroxide and water, what ends up happening is our other acid chloride hydrolyzes to form our carboxylic acid. And so what we've got here is essentially our nylon 6,6 dimer. And so this is essentially going from our first step where we've got all of our monomers, and then we begin to form these dimers. And over time, we're going to continue the polymerization reaction. The dimers will become trimers, will react with other dimers and trimers to form our nylon 6,6 polymer. So one of the first things that we would need to do coming into the lab is to flame dry some of our glassware. So we're going to need a 10 milliliter round bottom flask for our reaction, a 25 milliliter round bottom flask for our workup, our Claisen head adapter for our reaction, drying tube for our reaction, and a 25 or 30 milliliter beaker for polymerization. So we do have some moisture sensitive reactions that we are working with here, and we'll talk about why exactly that is, but it's very important that we prepare our glassware accordingly so that we don't run into any problems. So after we flame dry these, we want to cool them in a large beaker until the glassware is cool enough to be handled and our apparatus can be assembled. We want to make sure again that we're avoiding any contact with water after we have those flame dried. So the first step is going to be the synthesis of our adipoyl chloride. And so we're taking our adipic acid and we are reacting this with our thionyl chloride, SOCl2, the catalytic amount of DMF, dichloromethane, 50 degrees Celsius for 90 minutes. That's going to give us our acid chloride. Now it's really important that we convert this carboxylic acid into our acid chloride Because if we were to just take our carboxylic acid and try to react it with an amine, that amidation reaction is very slow. We tend to see an acid-base reaction take place between the amine and the carboxylic acid first, and so you have to really heat that up quite a lot. And so that's how they industrially make nylon 6,6, but for us practically in the lab, since we can't exactly get things to a super high temperature or under pressure, this is the method that we choose. So we are going to mix these together, and we are going to reflux this reaction for 90 minutes. We've got some other things that we need to do during that period of time. A couple of things to note. Adipic acid is sparingly soluble in dichloromethane, so when you put these things together, you will not see complete solubility. But as the adipic acid is converted into the adipoyl chloride, it becomes more soluble in dichloromethane. So we expect to see a change in observations here. So our acid is a solid, and as it is converted into the acyl chloride, the solid is going to disappear or dissolve because our solubility is changing as the structure is changing. Now we're adding in a catalytic amount of dimethylformamide, or DMF. So if it's catalytic, that means at the end of our mechanism it's going to be regenerated. But the role of the DMF is to activate our SOCl2. So let's take a look at what that mechanism looks like. So we've got our reaction scheme up top here. And we're going to start actually with our thionyl chloride. Now the Lewis structure for thionyl chloride is interesting because our sulfur atom actually has a lone pair on it. So sulfur and its ever-expanding octet. So here's our thionyl chloride. And this is going to react with our DMF. And so when we consider which atom of DMF is going to be our most nucleophilic, we would consider that our most nucleophilic atom is actually the oxygen atom. And if you can't convince yourself of that, it's a good idea to draw a resonance structure. So our sulfur atom of the thionyl chloride is our electrophile because that sulfur is attached to 3 very electronegative atoms. So our electrophile, or E+, is going to be our sulfur atom of our thionyl chloride, and our nucleophile is going to be our oxygen atom of our dimethylformamide. And so we can show this mechanism by using the lone pair of electrons on the nitrogen of our DMF to form a nitrogen-carbon double bond. Now when that happens, carbon is violating the octet, so we have to break a bond. The oxygen-carbon double bond is the easiest one for us to break. And so we show that those electrons are kind of coming up onto oxygen and being used to form a bond to sulfur. Now sulfur is going to have to break a bond. The easiest one for sulfur to break is going to be the sulfur-oxygen pi bond. And so ultimately what that's going to give us is a structure that looks something like this. We'll fill in all of our lone pairs as well. So it can be a little bit tricky here to make sure that we get everything attached in the proper way. Make sure all of our formal charges end up in the right spots. Don't add in any extra lone pairs or leave any off. All right, so now what we've got is this structure here. And so essentially what we learn in Chem 224 is that having a carbon-oxygen double bond is really, really thermodynamically favorable. The same is going to be true for this sulfur-oxygen double bond. And so we're doing an addition elimination mechanism. So we've done our addition here. Next, we're going to do our elimination. And so we are going to reform that sulfur-oxygen double bond. And then we are going to kick off our best leaving group. And so that, in this case, is going to be one of our chlorine atoms, which is going to leave as chloride. Okay, so now what we've got here looks something like that. And so at this point, we know that that nitrogen, that positively charged nitrogen atom, really wants some electrons. And so we can get that nitrogen atom some electrons by doing an addition of our chloride ion. So keep in mind the carbon atom here, this carbon atom, is going to be quite electrophilic. All right, so now what we've got, we've managed to somehow get that nitrogen atom some electrons. And so again, we've gone from having a carbon-nitrogen double bond, which is thermodynamically quite nice, to having a carbon-nitrogen single bond. And so what we can see happen, this was again our addition mechanism. Let me fill in over here that this was our elimination. So we've got a series of addition-elimination mechanisms here. And so our next step is going to be elimination. And so we are going to reform the carbon-nitrogen double bond. And when we do that, we've got to break a bond. The one that we're actually going to break here is the carbon-oxygen bond. We're going to use the electrons that were in that carbon-oxygen bond to form a double bond between sulfur and oxygen. Now, why would we do this? We do this because SO2, which is ultimately what we're going to generate from this elimination mechanism or decomposition, is a gas, and it's very thermodynamically stable. So we are forming the oxygen-sulfur double bond, and at that point we're violating our octet in ways that we can't do, so we have to break the sulfur-chlorine bond. And so we're going to get a couple of different products from this. We're actually going to get 3 different molecules. And so we have made what is called the Vilsmeier reagent, which is right here. And we've also made SO2, sulfur dioxide gas, which we put this arrow here to show that that's going to be expelled from the reaction. And we generate our chloride ion as well. So our Vilsmeier reagent is our chlorinating reagent. It is our halogenating reagent. And so this is what's going to react with our carboxylic acid. Now I'm going to abbreviate the structure of our carboxylic acid for the sake of space and for the sake of time. But again, we want to consider what atom is going to be our most electrophilic and what atom is going to be our most nucleophilic. So our most electrophilic atom is going to be the carbon here of what looks sort of like an imine or iminium. And then our nucleophilic atom is going to be this oxygen atom right here. So again, if you aren't convinced of that, be sure to draw some resonance structures. So this mechanism is going to look quite similar to what we saw earlier on. So let's go ahead and see what this product looks like. Sometimes it can be helpful in these types of situations to number your carbons, to fill all of those things in so that you don't lose anything or pick anything extra up. All right, so now we've got this sort of interesting looking intermediate here. And we've got an oxygen atom that has a positive charge, and we know that that oxygen atom with a positive charge wants electrons more than anything else. And so what actually takes place here is another addition reaction where our chloride ion is going to add to that very electrophilic carbon. I'll go ahead and fill these lone pairs in up here as well. And we're going to want to draw this in a very specific kind of way to make it look nice for our mechanism. And so I'm going to go ahead and take my time to make sure that I get this drawn exactly the way that I want it. So I'm going to go ahead and fill all of my lone pairs in now that I'm satisfied with everything being where it should be. So let's go ahead and move this down just a tiny bit. Just to make things a little bit easier on ourselves. Okay, so now what's going to happen is a very interesting type of process. And so where we start our arrows here is really not important. This is a concerted mechanism. This step of our mechanism is concerted. And so what happens ultimately is that we see that we reform the carbon-oxygen double bond that we have here. And that's going to essentially regenerate our DMF. Now we've got to break one of our carbon bonds that we have there. And so the carbon-chlorine bond is going to break, and the electrons that are in that bond are actually going to be used to remove this proton here. Now we've got to break a bond, and so the bond that's going to break is the hydrogen-oxygen bond. That's our only option, and that's going to reform our carbonyl. And so at the end of it, we've got our acyl chloride, or our acid chloride. We've got DMF. And we've also got HCl. Now that HCl is going to be kind of eaten up by the sodium hydroxide we have in our next step. We never really want to generate those strong acids, so we'll watch out for that. So while this mechanism is taking place, we're waiting that 90-minute reflux period. We're going to dissolve about 0.4 grams of our hexamethylene diamine in water and add in 3.5 milliliters of 2 molar sodium hydroxide. This should give us a homogeneous solution. And so this is one part of our reaction where we don't necessarily have to worry about water because we have water present. But one thing that I do want you to think about and consider is that we are taking this hexamethylene diamine and we're mixing it with sodium hydroxide. And so typically if we are talking about sodium hydroxide, we're talking about a base, we're talking about an acid-base reaction. So something I want you to think about is, does an acid-base reaction occur? And because we don't have any other clicker questions in this presentation, this is going to be your one clicker question for this presentation. So I want you to consider whether the acid-base reaction that occurs here is favorable. Does it occur to any appreciable extent? And why would our solution become uniform over time if it does or if it does not? So now we're going to talk about working up our adipoyl chloride. And so after our 90-minute reflux time, you want to make sure that you don't have any adipic acid left over, so no solids in your round bottom flask. You want to let that reaction mixture cool to room temperature for about 5 minutes. Transfer that to a flame-dried round bottom flask and remove any solvent using our rotary evaporators. That's going to end up giving you an oil or an oily residue in that round bottom flask, and we want to redissolve that in anhydrous dichloromethane. So again, here we want to avoid any contact or contamination with water, because when we have that acid chloride, that adipoyl chloride, it's potentially going to be able to hydrolyze to generate HCl gas, which is not good for us to be breathing in. So next we are going to look at our polymerization mechanism, and we're going to look at this just in one part. So we're not going to draw the huge polymer of nylon 6,6, but we're just going to look at what happens when we take these two reagents and mix them together. And so we'll start out by redrawing our adipoyl chloride. Whoops, too many lone pairs. Here we go. And so we are going to react this with our hexamethylene diamine. And so we've got a good nucleophile and we've got a very strong electrophile. And so we're going to react those two with one another. We'll go ahead and take a look at what those products look like. It's again a good idea to count your carbons, make sure you've got everything right before you move much further. All right, so we'll fill in our formal charges. And now we've got, again, an addition mechanism that's taking place here. And that's usually going to be followed by our elimination mechanism. And so what we see here is a tetrahedral intermediate, which has kind of a strong acid and a strong base both present. So one thing that some mechanisms might propose is a proton transfer reaction. But what actually takes place here, or what we can propose takes place, is reformation of the carbonyl, the carbon-oxygen double bond, and loss of our chloride ion. Now, thermodynamically, that's going to be very stable. Again, we want to make sure that we have the right number of carbons in our final structures. So we've got an ammonium species here, and we've also got our chloride ion that we just kicked out in our elimination step. Now the only thing that's left for us to get to our final dimer product is to react this with our sodium hydroxide. And so this is going to be a very fast acid-base reaction, quite favorable here. Let's make that look a little prettier. I think I left off a couple of carbons. Nope, we are all good. And so one thing that might be a good idea for us to do is to kind of keep track of those carbons, where they came from. And we can do that. You can go back and you can number the carbons of your starting materials. One thing you might notice is that we use sodium hydroxide here as our base and not chloride, since it's a stronger base. But we can go back and we can number our starting materials if we want to, just to make sure everything kind of matches up. We haven't lost anything anywhere along the way. So let's go ahead and number the carbons here. I'm going to give them prime symbols. They're different colors as well, but at least we can distinguish them. More ways than one, that's not going to hurt us. Okay, so we've got the right number of carbons. This is good. Okay, so we've got our polymerization mechanism that's taking place. And so we're combining our two solutions together. It's important to note that when we are doing this combination, we've got our organic solution in the beaker first. This is going to be our lower layer. So we're transferring that using a pipette. And then we are going to slowly add our aqueous solution, which contains our hexamethylene diamine, to the top. So that's going to be our upper layer. We want to add this down the sides of the flask slowly. Our polymer is then going to begin to form at our liquid-liquid interface, and so that's going to look kind of just like a film. We're going to use our stirring rod to pull and wrap our nylon 6,6 polymer. So the polymerization is going to continue to happen until we run out of our solution, one or both of our solutions. We're then going to rinse our polymer with deionized water. At that point, we'll take our nylon 6,6 polymer and we will dye it. So we're going to use that azo dye that we saved. We're going to take about 50 milligrams of that in about 50 milliliters of deionized water, warm that up so that our azo dye is mostly dissolved, so just a gentle warming, and then we're going to heat it up with our nylon 6,6 to 90 to 100 degrees. Toss our nylon 6,6 into the dye solution for 5 minutes, remove it, rinse it with tap water, and then dry it with paper towels. And then you'll have a couple of very vibrant nylon polymers. We do have a couple of important safety notes here. And so thionyl chloride is corrosive. Again, if it comes into contact with water, either moisture in the air or in your glassware, it will release HCl gas, which is not very good for us to be breathing in. So really important for us to handle that with care. Dichloromethane is carcinogenic. It has fast breakthrough time. So if you suspect that you have had dichloromethane come into contact with your gloves, you want to make sure you change those quite quickly. We're working with 2 molar sodium hydroxide, which is caustic. So again, if you get this onto your skin, you want to make sure that you rinse with water for a long period of time, usually at least 10 minutes. Some tips for you: if you have any thionyl chloride or in the adipoyl chloride steps, we want to make sure that we are avoiding water. And if we do have water present, we have to be aware that we could have some hydrogen chloride gas that is evolving into the air. So we want to always make sure we work well under our hood. As far as time management goes, we have a 90-minute reflux time, so prep other solutions, use that time to work on other things like possibly writing your experimental narrative. As far as waste disposal goes, all of our organics and organic contaminated aqueous liquids should go into acetone waste. And so organic contaminated aqueous liquids, we're talking anything that came into contact with an organic solvent. So even if you have an aqueous layer, if that was in contact with an organic layer, if that was in contact with dichloromethane, for example, that needs to go into the acetone waste. And then your nylon 6,6 polymer can be disposed of in the normal trash. So with that, we wrap up experiment 8, and we will talk again about experiment 9 next week.